it will say, really just pay attention to what I'm covering in PowerPoint. And there's a little bit of reading, but you're going to see that the chapter goes into more depth than what I'm going to do. All right, so really use the PowerPoints as a guide to the depth of information and amount of information, especially with the light reactions. So this took hundreds of years to figure out, and so it's been a long process. But when you think about photosynthesis, when the plant is running the light reaction, basically when you look at the whole thing, it's quite complicated, but when you look at it, it's like, okay, why is the plant doing this? The big payoff, one of the big payoffs, is to get ATP. And ATP is what all living organisms on Earth use as their chemical energy source. It's primarily ATP. There's some other ones too. It's not the only one, but that's the dominant one. And so that is a huge payoff when the plant runs the light reaction, getting that ATP. And it's actually quite remarkable how the plants have evolved to capture and absorb sunlight energy and turn that into ATP through this very interesting process. So now we're zooming in to the chloroplast of the cells. Typically, we talk about leaf tissue, leaf cells, but remember, there's some plants that don't really have true leaves, and it's their stems that do photosynthesis. So there's some variations on where you're going to see this happening. But basically, if you've had a little bit of training in photosynthesis, this is the equation they like to show you that gives a synopsis of both actually the light reactions and the biochemical reactions all put together. And so this is like a huge umbrella equation. And so what they're doing here is they're saying, okay, the plant will take in CO2 and there's going to be water involved. And then light has to be a part of this process. And I usually, over the arrow, I'll put in chlorophyll. You can't forget the pigment. If the plant doesn't have chlorophyll, this isn't going to work. And then you see these products of C6H12O6 and water. So when I say C6H12O6, what word pops into your head? Sugar? Is there a specific sugar that you like to think about? Glucose? That's what I like to. I mean, that's just like trained into me. Glucose, C6H12O6. So I don't, when I teach upper level plant phys, I give them a slightly different equation because it's actually not glucose. That's the first sugar that the plant makes. That's not what they do. And so it's a little bit confusing, but I understand why they write it this way. And so, I'm just trying to orient your head if you've seen this equation before, but actually glucose isn't the first sugar that they make. C6H12O6 is just referring to it's a sugar. And they're trying to, so you have to keep it a little bit more vague, a sugar, okay? So this equation is a massive summary statement. What you're going to see is that there's a whole lot more to it than this. And so there's lots of intermediate steps. And it's a very complicated process when you look at how the plant manages to absorb that light energy and convert that into ATP. So it's fascinating to see, and it's super efficient, really, when you think about it, and quite amazing. Wouldn't that be nice if we could do that? Just go to the beach, lay out in the sun for 15 minutes and be full, have all of our nutritional needs met pretty much. So this picture is, again, a big picture statement. This is like putting it all together. This is photosynthesis. And so we see on the left here, you see light striking the, what is the inner membrane, the very innermost membrane that's floating around inside the chloroplast called? Do you remember? Thylakoid. Right, so the thylakoid membrane is where the chlorophyll is embedded. The light energy is being absorbed by that wonderful chlorophyll. And you can see one of the payoffs is ATP. Another big payoff that we'll get into is NADPH. That's another big one, all right? And then you see this arrow linking over to the right, where you see this circular pathway. So this is a cycle pathway. And you can see that this is where we see the CO2 that the plant takes in through its stomata will go into this Calvin cycle. All right, so we're going to be focusing a lot on once the plant makes ATP and NADPH, how do they do that? How do they run the Calvin cycle? All right, so we'll be looking at that. And then you can see the payoff from the Calvin cycle is those lovely sugars, okay? So when we think about the light reactions, we're zooming in to the thylakoid. So if you cracked open the chloroplast and looked inside, you would see that internal membrane floating around. And it's in the thylakoid that you have all that chlorophyll pigment embedded in it. And in regions of the thylakoid, you're going to see that the plant has this amazing capability to take water, H2O, which is such a stable molecule, and extract electrons from that. That is incredibly difficult to do. And so the plant can do it in short order. And it's a very special part of the chlorophyll molecule, or I mean the thylakoid membrane, where you see this happening. As those electrons are ripped off of water, then those electrons get fed into an electron transport chain, where the electrons get passed from a donor to a recipient, donor to recipient, on down the chain. And you have this big, long reaction pathway. And as that is happening, it creates a situation that allows for ATP to be formed. And so this electron transport chain process goes hand in hand with ATP production. So it's a huge payoff. And so ultimately, when you look at photosynthesis of how higher plants do it, they're extracting electrons off of a very stable water. And we have them to thank, because as they're doing that, as you keep stripping electrons off of water, eventually you're going to have oxygen as a byproduct. And so that's how we get oxygen building up in our atmosphere over millions and millions of years, is the plants doing this. And as they, so when we talk about the light reactions, we really don't focus too much on oxygen. I mean, it's great for us. It allowed our species to evolve and live on this earth. But when you look at the light reactions, the plant really doesn't care so much about the oxygen. They actually get rid of that for the most part. They're focused on, I want those electrons from water, and I want to take those electrons and run it through this pathway, this electron transport chain process. Because if I do that, I can create a situation where ATP can get synthesized. And so that's the huge payoff. It's to get that wonderful ATP. And that's needed so that they can do part 2 of photosynthesis, biocatalysis, to get those sugars produced. So as they run this electron transport process, that electron has to end up somewhere. Someone has to be the last acceptor. When you look at any electron transport chain process, there's a final or terminal electron acceptor that will, it stops there, okay? And so that is where we're going to see that NADP, you should actually put a plus after the P, NADP plus is at the very end of the electron transport chain. And so NADP plus is sitting there going, I'm waiting for my electron. And when it gets that electron, it will form NADPH. And that's really good, because that is also needed in the biochemical reaction to get the Calvin cycle to run, get your sugars. So 2 huge things happen during the light reaction. You get that ATP produced. Nicotine amide adenine. So big long words. Very, very long word. And it's great because it's useful. NADPH is useful because we're going to see as it gets used over in biochemical reactions, Calvin cycle, it's really good at donating. So we call it a reducing molecule. If it's a reducing molecule, donate electrons where it's needed. And so that helps keep the Calvin cycle going. And so what I've described here in a few bulleted word sentences is being shown here. Again, not all the details are being shown, but this is the one that I show I start with. You work through this in more detail than actually what's shown here. So I'm not going to ask you to know this to any level of detail, other than really what we see happening in the previous slide. That's the level of detail I'm thinking about in terms of what's going on in the light reactions. I want you to get the big picture idea and the big picture process and what is the payoff. Why is the plant doing this? All right. If you take plant phys, you'll get into all the amazing, wonderful bits and pieces that how a plant puts this all together. It's just phenomenal. But what I put this here is if you're a visual learner, I just want to get you thinking about this idea of water is over here on the lower left. And this is a complex that's capable of ripping electrons off of water, extracting those electrons. And then these black dotted arrows are it's showing the pathway of the electron transport chain. So the electrons are getting passed from player to player in this chain. And then the very end acceptor is NADP+. So we call that the terminal electron acceptor or final electron acceptor. And when it takes that electron, it will also grab a proton, and then it becomes NADPH. And now we say it's reduced because it got that electron. And so it's a reducing molecule. Now it can go over into the Kelvin cycle and say, hey, I can donate an electron, keep this going. So you see this is oriented into we're inside the chloroplast here. Use this membrane that they've drawn. That's the thylakoid membrane. All right. And then the stroma portion of the chloroplast, that's just the watery portion. If you were to crack open the chloroplast and look inside all that watery goop, we call that region the stroma. And so it's really where it's the stroma where you see NADPH getting made and ATP. Okay, so you also see that, and again, I won't go into huge detail, but you can see here that what they've drawn here is that the electron flow is happening all the way to NADP+. And then they're also showing that in that process, you create conditions that allow for ATP synthesis. It's through a development of a proton gradient, we call it, because what's happening is as you strip electrons off water, you get oxygen, but you also get protons as one of the products. And so as you have that happening, and then there's a few other places here where you get protons building up as this whole thing keeps cooking along. And then as these protons diffuse through the ATP synthase, it actually allows for the ATP synthase to make ATP. So it's amazing. This ATP synthase is actually, they refer to it as a molecular motor because it actually has a torque force movement. It will actually rotate those funky looking balls that you see here. Those will actually rotate and allow for ADP and this phosphate to react with each other. And when that happens, you end up with that wonderful ATP. So very, very complicated, but I want you to zoom out and think about the big picture idea. Other questions on the light reaction? Yeah, if it weren't for them moving, none of this would happen. And so as you get into more science classes, you'll see this is a common theme, whether you're talking about plants, animals, bacteria, fungi, you're going to see that they all have these methods. Let's put an electron transfer chain process going, because as I'm doing that, I can create a proton. And if I can create a proton, I So it's all linked together. You can't really separate. And it all kicks off with this electron transport chain process. And so it's kind of a universal theme. We're doing that right now in our mitochondria. This is a similar, well, unfortunately, we can't absorb light energy, but we also have an electron transport chain going right now. If you ate a good breakfast, especially, you have that cooking along and you're making all kinds of ATP in your mitochondria. Other questions on the light reactions? Okay, spend some time with this, thinking about it and working through it. When I learned this, even though I'm just asking for a big picture idea, you can still draw out like the idea of an electron flow, the idea of protons building up. That makes ATP happen, right? What else? You know, the NADPH. So talk it out, draw it out, give it some time to sink into your long-term memory. So once the plant has made that ATP and the NADPH, that's required in order to push the biochemical reactions. And so with the biochemical reactions, we are zooming into the chloroplast still, but we're looking at the watery portion, the stroma, right? The stroma of the chloroplast. And so that's where the plant is going to run so-called Calvin cycle. It's right there. And the Calvin cycle, we're going to see, needs an input of ATP and NADPH in order to take it to completion. So the reason it's called the Calvin cycle, whoops, what happened there? The reason it's called the Calvin cycle is it's named after Dr. Calvin, whose full name is Melvin Calvin. And he is credited with kind of breaking open the, they were at a spot of learning about the Calvin cycle where they couldn't figure out a key thing. They just couldn't figure it out. And he came along and he kind of looked at it from a different perspective and just opened it up wide, and they find everything started to fall into place. So his contribution, he actually was awarded the Nobel Prize. And if you are from the Houghton area, Keweenaw area, he went to University as an undergrad. So when I was there doing my PhD work, Melvin Calvin was a big deal in the biology department because of that. So he is the one given the major credit. There are often sometimes with the other names associated, like Photosynthesis cycle. So there are other scientists too, but I tend to shorten it down to Calvin. So that's how I refer to it. So this is the kickoff to the Calvin cycle. This is how it starts. The plant will open up its stomata, take in carbon dioxide, and then it reacts it with this molecule that we abbreviate down to RUBP. And I'll show you that in its full designation in a minute. And then eventually you're going to get sugars from that. So that's the kickoff of it. And at key points, we're going to see the ATP needs to help out and NADPH has to help out. And those came from the light reactions. RXN is my way to abbreviate reaction, okay, the word reaction. So when they were initially, before Calvin came on the scene to this, they knew that when they were tracking the Calvin cycle, they said, okay, we know that the first stable molecule is 3 carbon. So they're like, okay, and we know that CO2 is involved. They knew that much. They knew that CO2 has to come in and enter into the Calvin cycle. They see that the first stable molecule is 3 carbons. So they're like, okay, well, carbon dioxide has 1 carbon. So we're looking for 2 carbons that it can react with. So you end up with that 3 carbon. So they were hunting for a 2 carbon molecule that CO2 reacts with, and coming up empty, nothing. And so then Calvin, one of his major contributions was he came in and said, well, there's more than one way to get to 3. What if you took in CO2 that was unstable? They knew CO2 was a sulfur. So they said, okay, there's another way to get to 3. What if you take a pipe molecule, react CO2 with it? Now you're up to 6. And then if you divide that by 2, then you're at 3. And that's another way to get to 3, right? it was quite ingenious, you know, thinking out of the box, so to speak. But that was like the huge light bulb moment. Like, oh, yeah. And so then they were able to piece it together that it's RUBP that is the molecule the CO2 reacts with. And RUBP is 5 carbon, the 5 carbon sugar. And so here you see it, and here it is all spelled out. Ribulose 1,5-bis phosphate. Only forgot it's usually bis, not biphosphate, bisphosphate. So it has 2 phosphates, and they're showing that on either end. And then all the gray circles or spheres, or those are the carbon. All right, so it's a 5 carbon with 2 phosphates. And so rather than spelling that out all the time, we just abbreviated RUBP. Okay. And so you can see RUBP is sitting there, and then up in the upper right, you see carbon dioxide. So carbon dioxide and RUBP react. And then in the pink here, you see this word Rubisco. That's the enzyme. All right, the enzyme. So you have to imagine this enzyme has an active site, and it's sitting there, and it will take RUBP in, CO2 in, react them, and you get this 6 carbon molecule. Back in the day, they never saw this 6 carbon molecule because it's so short-lived. They didn't have the technology to pick up on it, that it was actually being made. Finally, the technology caught up, and they realized, oh, there it is. There's that 6 carbon, but it doesn't live very long. It quickly breaks down into 2 identical PGA molecules. All right, so that's what you see here. So that whole step there, that took a long time to figure out, partly because the technology just wasn't there to pick up on that 6 carbon. Okay, so that first portion of the Calvin cycle where you see the CO2 and RUBP reacting, nowhere in there do you see anything that says anything about ATP or NADPH. So what that's saying is, if the plant can take in CO2 and if it has enough RUBP and those two are around each other and dump into Rubisco's active site, it's just going to happen, spontaneous. You don't need any extra ATP for that. You don't need NADPH. It's just going to happen. And so that first step is quite easy for the plant. And then you get these 2 identical PGA molecules. But then you do see ATP coming in. All right, so what's happening here is these 2 PGA molecules will each react with an ATP in order to form the bis-phospho glyceric acid, or glycerate. If you see glycerate, ATE, on the end of a word, that means it's an acid. So it can be said as glycerate or glyceric acid. Either one is correct. And so when I teach this in plant physiology, that's interesting. I just noticed that they did a different abbreviation. So PGA is usually how we abbreviate phosphoglyceric acid. And then when it comes to, whoops. Oh, there it is. Right there, G3P. I thought, wow, that's weird. They did it wrong. They didn't. All right, so here we have the PGA with the help of ATP is going to get converted to glyceraldehyde 3-phosphate. This is one of the intermediates, the being shown here at the very bottom, which I don't really focus on. I talk more about, okay, you have this PGA molecule, you have ATP, and then the other step here that's really important is the NADPH is coming in. So this second phase of the Calvin cycle, that's where you start seeing ATP is needed and NADPH is needed in order to push this reaction from PGA to the G3P, glyceraldehyde 3-phosphate. So if all of that goes okay, what happens is the plant is doing this over and over and over all day long. as long as the stomata are open and they're taking in CO2. So after a while, you're going to get a buildup of this G3P. Everything's going okay. And then you see this little arrow going off of the cycle. So what they're trying to show here is that this is a point where some of the G3P can get diverted into another pathway in order to make the final sugar, which actually is sucrose. The plant's goal is to make sucrose, not glucose, right? So this G3P that starts to build up during the day, they'll take some of that and run it into another pathway that will make sucrose. And that's great for the plant. But the other key thing about the Calvin cycle is that it's a cycle. You have to get more RUBP made all day long so that as the plant continues to take in CO2, it has the RUBP to react with. If you don't keep up making that RUBP, then when the CO2 comes in, there's nothing for the CO2 to react. So you have to keep enough of this RUBP being made all day long. See this, that's why you see it's this cycle. And then again, if you follow this last portion of the cycle, you see that G3P will, and then I like how they put this arrow, arrow, arrow. That's the artist way of saying there's a lot happening there that they've glossed over. Okay, so arrow, 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 and then you finally see this product here, and then that is another place where ATP is needed in order to get your final product of the cycle, which is RUBP. So lots of stuff happening here. Key points where we see ATP and NADPH. And then we see that there is this kind of branch point here where some of that G3P gets sent into another pathway in order to make sucrose. So if you look at the full thing, this is the same picture we just looked at. I just revealed this other, here they're giving you details of the blow up here. So here they have it where you can count carbons. The one down here is more just words. Okay, so it's for studying purposes. And then you can still see that there's this branching point where the G3P can funnel into a different pathway. So the Calvin cycle, really, when you just look at the Calvin cycle, you don't see sucrose being made in here. It's the idea that this G3P goes into a separate pathway, and then that creates sucrose. Okay. But you have to have that Calvin cycle cooking all day long in order to get lots of that G3P that can get sent into that other pathway to make sucrose. So there's another diagram here that I put into the slide set. This is not from your book. It's just for study purposes. It's basically saying the same thing we just went through. It just has some more words on here, little different orientation. So if it helps you study, great. But basically, it's saying the same thing as this. Okay. I'm going to go back to this one. All right. Questions on the Calvin cycle? I'm guessing you haven't learned about this in any other class, unless you take the class. Which is great. Okay. So when you're studying that, the key points I want you to be thinking about is that what kicks it off? What starts the Calvin cycle? It's the CO2 coming in. Well, how does that happen? Well, the plant has to open its stomata, right? And so during the day, this can only happen during the day, by the way. So the plant can't make sugars at night. They don't run the Calvin cycle at night. And I get more into this plant bit, but the reason for that is some of the enzymes involved in the plant, they have to be fully functional. So this only happens during the day. So as the sun strikes the plant, hopefully they're gonna water in the soil where they can risk opening their up, take in that CO2 and immediately react it with RUBP. This enzyme is Rubisco. So please start studying this as soon as you can and get straight what RUBP is versus what is Rubisco. Totally different. Even though I've seen students mix those up previous semester. So please, every applicant. Okay. RUBP is the five carbon sugar. Rubisco is the enzyme. And Rubisco is an active. The C on the end of the word toward the end there stands for carboxylase. So if you are a carboxylase enzyme, that means you can take in CO2 into your active site. The O on the very end of the word is oxygen. They both those words end in ASE. Anytime you see ASE in science, think about an enzyme. So that much you remember, we have Rubisco enzyme. Carboxylase, oxygen. So the O in oxygenase means that this enzyme is, I like to say, sloppy, or it's very like easygoing because it can take CO2 into its active site. And it's like, yeah, I can do that, and I can react CO2 with RUBP. But it can also take in oxygen into its active site. And so what they've discovered is it's whatever Rubisco has more around itself. So if it has lots of CO2 around it, that's what wins, comes into the active site. That will react with RUBP and you'll get that 6 carbon. But if oxygen is more abundant around Rubisco, then Rubisco's like, yeah, all right, fine. And it will react oxygen RUBP. And that's not good. We're going to talk quite a bit about that. But first, let's continue on. Let's just think about this Calvin cycle. So if everything's going okay, CO2 comes into Rubisco's active site and reacts that with RUBP, a 5 carbon sugar. You get that 6 carbon short-lived intermediate. I don't care that you know the name of it, but the big point is it's going to break down into 2 identical molecules of PGA. So being able to recognize that that's the first stage of the Calvin cycle. So we'll think about that as you're studying. And then the second phase is where you start to see ATP and NADPH. It's taking PGA and getting the reaction pathway all the way to G3P. You don't need to know the intermediate. PGA to G3P is what I need you to know. And as you take PGA and work the pathway to G3P, you do see that you need ATP and NADPH, and that those are coming from the light reactions. Okay, so that's like phase 2 of the Calvin cycle. And then the last phase is the biggest phase that they've abbreviated here, where you take G3P and end back up with RUBP. And all of these arrows here that you see being drawn, all of these arrows here, there's a lot happening there. And we get into that plant phase, but we won't do that here. All right, so there's a lot more stuff happening there. But basically that third part is taking G3P and making more RUBP. And you need ATP there too. So usually if I ask you questions on, this won't be obviously on today's exam, it's going to be on exam 2. I tend to use the abbreviations. So I encourage you to learn it that way. RUBP, Rubisco, PGA, G3P. So learn it that way, I think, is the best way to do it. Question on the Calvin cycle. Clear as mud? Especially if this is your first time hearing it, yeah, it's going to be like a lot to take in. And I get that. That's why you need to study this more than once, many more times than once. Remember, repetitive studying really does convert things from, right now everything's in your short-term memory, but now you got to convert it into your long. Way to do that is hit it, every day. Short first.
So just confirming, just concept, you don't do it like very well.
No, not really labeling, but like knowing how it kicks off, the basic big picture idea of the site. Yeah, and know that yes, it in any stage are needed at this stage, Other questions on the Calvin cycle? All right, so now we need to think about Rubisco a little bit more. Now, Rubisco evolved actually back when bacteria started doing photosynthesis. So we're talking a couple billion years ago. Well, back then, our atmosphere had no oxygen. So Rubisco never really had to deal with oxygen when it first evolved. It only cared and worked with CO2 because that's the only thing that was around. Oxygen wasn't there yet. But then eventually the cyanobacteria came along and evolved. And cyanobacteria are capable of doing photosynthesis, very similar to us, and they give off oxygen. So now, over tens of millions of years, as cyanobacteria are doing, our oxygen levels kept building in the atmosphere. But Rubisco never really adapted to that. And so now, in our current world, where we have 20% oxygen in the air, Rubisco can't really tell the difference between CO2 and O2. It doesn't discriminate between the two and will let either one come in to its active site and react with that RUBP. And then if that happens, it creates a little bit of a dilemma or problem for the plant because it creates products that can't feed directly into the Calvin cycle. So it's kind of a mess for the plant. And as scientists were studying this and running some careful experiments in a lab environment, like that. Hang on. As they were doing that, they realized that what they would do is they take these plants and they make oxygen really available inside the plant, CO2 or very low levels, and they'd watch the plant do this other pathway, and then they watch the plant. It wouldn't grow as fast. It would form flowers, and so they said, okay, too much, and there's oxygen winning coming into the visceral network. Plant seems to suffer lower yields, lower growth rate. So it's probably not the best thing the plant to do on a regular basis. And so the lab experiments kind of forced a situation of let's make the plant only see the oxygen and see what happens. And they realized it was detrimental to the plant. So basically what they were doing is they were feeding the plant a lot of oxygen and not much CO2. And so Rubisco kept taking oxygen. And so that's the oxygenated aspect of this plant, able to take in oxygen into its active site. And we say that carbon dioxide and oxygen compete for Rubisco's active site. They're duking it out. Whatever's more abundant around Rubisco, that's who wins. So if there's lots of CO2, just a little bit of O2, CO2 comes into the active site. And then you get the right conditions for the Calvin cycle to start cooking along really nice. But if there's more oxygen compared to carbon dioxide around Rubisco, then you have RUBP reacting with oxygen. And now you end up with a situation where you get one PGA as a product, which is good. That can immediately go into the Calvin cycle. That's fine. You feed into that part of the Calvin cycle. But then you get this other funky molecule called phosphoglycolate. That's a 2-carbon molecule. That can't feed into the Calvin cycle. It just can't. There's no enzymes in the Calvin cycle that can easily take that 2-carbon and just snip it into the Calvin cycle. It doesn't work. So the plant has evolved this mechanism where they take that phosphoglycolate and they say, okay, I can immediately slip it into the Calvin cycle, but I'm going to evolve a mechanism where I can eventually create a molecule that can fit into the Calvin cycle. And so what they do when they study this other cycle that this phosphoglycolate gets fed into, they know that it does help the plant create a molecule that can feed in. So that's where they say it allows for some partial recovery of carbon. What they mean by that is, in the end, there is a molecule made that can feed right into the Calvin cycle. So that's good. But in this process, there's a loss of carbon we're going to see. So some carbon is lost in the sense that, weird, because as you look at this other pathway, carbon dioxide is actually made. Instead of being taken and combined with RUBP, it's actually made and then given off. And so they say it's lost. So we'll look at that. And then another fancy word for this pathway, they refer to it as photorespiration. And what they're saying there is, like, we're all carrying out respiration. When we exhale, we're giving off CO2. And when the plant makes CO2 during this pathway, that's, respiration. So that's why they call it photorespiration. So this is all in words. Let's look at it as kind of a flow chart. So again, this is saying basically the same thing as the previous slide. We're just kind of looking at like a decision point. So here you have RUBP, and if it has high CO2 levels and low O2 around Rubisco, you get branching off to the right here, where you get those two PGA molecules. It'll keep going through the Calvin cycle. Everything's great. On the other hand, if Rubisco is seeing low CO2 and higher oxygen, then Rubisco takes in oxygen. And that's when you get this one PGA, which is good. That can go right into the Calvin cycle, but that phosphoglycolate, darn it, it's a 2 carbon. There's no way to fit it in to the Calvin cycle. And so that gets diverted to a totally different pathway. And it is a really big pathway. So, and then you see arrow, arrow, arrow. Again, they're trying to say there's a lot happening here. All right. And then you see CO2 is being given off. So that's where they're saying it's a loss of carbon. And that just, disappears, doesn't enter into the Calvin cycle. So here's another way to look at it. If you like to count carbons and see the actual chemical nature of this reaction. Here you see RUBP on the left reacting with oxygen. And you can see that there's one PGA produced, which is great. But then you get that funky 2 carbon phosphoglycolate. That phosphoglycolate can't go into the Calvin cycle. Instead, it goes into this cycle. Oh my goodness, look at that thing. It's a monster. So what we see here is on the left, it's the same thing on the right. It's just 2 different artists' rendition of it. So when I teach plant phys, I teach off the right one because it's a little cleaner. It's just easier to look at. But the one on the left, you can count carbons and look at the more detailed version. Now, we're not getting into the details in this class, all right? I put this in here to impress on you. It's a big pathway. So some of the things I want you to know for this pathway is that the plant has to coordinate this pathway through 3 different organelles. So I want you to know those organelles. It starts off in the chloroplast, and then some of the intermediates have to find the peroxisome next and do the next phase. And then after that, the intermediates have to go into the mitochondria of the plant and do the next part. And not only that, this whole thing has to run twice in order to end up with this very last, very precious molecule here. That's PGA. And then I drew the C3 cycle there. So that PGA, once it finally gets made, it can get dumped into the C3. Yes. Yeah, and then they lose CO2 as carbon dioxide, so it's not as efficient for sure. Yeah. Yeah, you still get a PTA, one though, just one. Okay, so, all right, we'll continue with this next week. I'll do a little bit of review, and then we're gonna find out that because of rubisco being so way back about what it takes into its active site, that's allowed or forced plants to come up with different strategies on how we make the. We're gonna link those ideas together. So we'll get into that next Tuesday. And remember, I'm coming into each of your lab sections, so I'll be in there about 1:15 for the first one. So do the multiple choice lecture first so that I can be available to answer questions for you and for the exam today.